Hello, everybody. In this lecture, we are going to talk about prokaryotic or really general membrane transport. But before we get right into it, I wanted to point out that I used AI to make this image, which is supposed to be a cell membrane. I thought it turned out kind of okay. So with that, we are going to now get right into it. So by the end of this module, you should be able to differentiate between active and passive transport, define osmosis, and predict the outcome of placing a bacterial cell into hypertonic, hypotonic, or isotonic solutions, and identify the types of passive and active transport mechanisms that exist in cells. And so we have active and passive transport. The basic way of differentiating that is that passive transport does not require any energy, but active transport does. So here on the active transport side, we see our ATP. Also, you're going to, with passive transport, have a movement from high concentration to low concentration. While with active transport, you're going to usually go from low to high concentration, but there can be some other active transport as well. Now, our simplest form of passive transport is going to be diffusion, which is going to be passively moving from high concentration to low concentration. So when you look at this image, you should see that we have higher concentration on this side of the cell than we do on this side of the cell. And so we're going to expect these blue bubbles, whatever they're representing, to diffuse through the cell to this side. Now, with simple diffusion, they do that by going right through the plasma membrane, right? So we're going to have outside of the cell, through the plasma membrane to the inside of the cell. Now over here, we have a different molecule, the red molecule. It's still going to move from high to low, but for whatever reason, this molecule cannot pass through the plasma membrane. So it needs to be helped. And so in fancy science terms, when we're helping with diffusion, we're facilitating that diffusion. So here we have facilitated diffusion, which means that it's going to be transporting through some sort of protein channel. In this case, we have a transport protein. And so the things that are going to work for your simple diffusion are going to be your small non-charged molecules. It's going to be your gases, things that are lipid soluble. They can just go right in and out. Facilitated diffusion, still going to move along that gradient, but these are going to potentially be charged or polar molecules, things that are not uh, easily soluble, and they're going to require some sort of channel protein, something that something that's transmembrane that goes across from the outside to the inside, our channel or carrier proteins. And so we have a couple of different examples that we can see here. We have a non-specific transporter. Over here, there's a specific transporter. So this is just going to let whatever can fit through. This is going to require a specific molecule to basically trigger it to open and release it. And then here we have an aquaporin, which is for osmosis. Well, sorry, it's in addition to osmosis for letting water through. All right, so we should talk a little bit about osmosis, which is really just a special type of diffusion, which is specific to water. And so diffusion of water or osmosis functions the same way as diffusion of these other things. It's going to flow from, it's basically trying to equilibrate your uh, your concentration of inside the cell and outside of the cell, and of your solutes, right? So of any of your salts or anything else that's dissolved in that water. And so if a solution is isotonic, iso in this case is going to mean equal. Um, What that means is that the concentration of solutes outside of the cell, which is represented by these little pink dots, purple dots, is equivalent to the concentration inside the cell. And if you have an isotonic solution, then water doesn't flow. It just stays. It doesn't move in and out. It doesn't diffuse or it doesn't osmose. However, if that concentration gradient is different on one side or the other, then water is going to move in or out of the cell. So in a hypertonic environment, that would be something that is got more solute dissolved in it than the inside of the cell. And so think of like the ocean or the Dead Sea or a glass of salt water or something along those lines. Of course, when we're talking about the environment, this can be on a large scale, like those environments, or they can be micro scale, where there are areas where there are hypertonic environments. And so since there are more solute outside of the cell, the water wants to leave the cell to equilibrate. And when it does that, it's going to actually cause plasmolysis. And in that process, the cell wall can actually um, dissociate and pull away from the membrane. And you get this you can basically get this shriveled up cell. Now, on the other hand, if you have a hypotonic solution, so if you take a cell and drop it into pure water with no solute in it, then you're going to have a hypotonic environment. And that means that there's more solute inside the cell than outside of the cell. And now all that water that is inside, or sorry, all that water that's outside the cell wants to get inside the cell to dilute all those solutes. And so the water will come in until it equilibrates, or potentially until it bursts the cell through a process of lysis. All right. And so here's just a single image showing that isotonic where we see that there's no net change in water inside or outside the cell. Here's hypertonic where we see that the outside of the cell has more solute and therefore the water leaves the cell, leading to this plasmolysis. And then we've got a hypotonic solution where there's more solute inside of the cell, and therefore the water travels into the cell, uh, causing the cell to potentially lyse. All right, so moving into active transport, this is something that requires energy to transport substances through these channel or carrier proteins. And so active transport can move things along the concentration gradient or against the concentration gradient, while diffusion can only move along the concentration gradient. And here we have a couple different examples of our different active transports. We have primary, secondary active transport, and then we have the phosphotransferase system. And so in this primary active transport, we just have a sodium potassium pump where really all you're doing is just transporting sodium outside of the cell and then potassium inside of the cell. And in doing this, you actually need ATP. Now, then there are these secondary um, active transport systems. And here, what you do is it's kind of like it's like a co-transport in this sodium glucose channel, right? So we have this, in this case, we have a symporter, which means that they're both going in the same direction. And the sodium is going to be pumped into the cell along its concentration gradient. Um, and that actually allows to fuel glucose to be pumped into the cell against its concentration gradient. And then we have phosphotransferase systems, um, and they are going to have some sort of intermediate like PEP, phosphophenol pyruvate, phosphorylate. So what they, sorry, they're phosphophenol pyruvate, and what they do is they phosphorylate. So they add these phosphoruses to the molecules that's being transported. And so this is like what actually happens in like the first step of glycolysis. When you transport glucose into the cell to enter glycolysis, oftentimes um, you're going to get a phosphorylated glucose. And several of these systems are going to use ATP, like in your different primary active transport systems. And so what that means is that you actually need to have produced ATP to fuel this. And probably the most common one is the sodium potassium pump, where sodium goes from high concentration uh, outside of the cell to inside of the cell, and potassium goes um, the opposite direction. Uh, in terms of they're if we look here, we see I said it backwards there, that the sodium is actually going out of the cell, and then the potassium is coming into the cell. And then when we look at our secondary active transport, this is the ion gradient. And so when you are able to transport, this is not the best picture for this one. When you transport, and when we go back to this one, it's a better picture. Um, we're basically transporting the uh, ion along its gradient to fuel the ability to transport the molecule of interest, in this case, glucose. And so with the secondary active transport, this is going to be some sort of co-transport system, and these come in two forms, symporter and antiporter. So symporters are going to both be transported in at the same time, and antiporters are going to transport one in and then one out. And so these are also the same sorts of things that you might see with an antibiotic efflux pump. And so here we have an example of the phosphotransferase system, which you by no means need to know all of this, but this is just kind of reiterating the idea that as that sugar is coming in, it's going to get this P. You see this P kind of moving along this chain, and then it ends up on the sugar. And so that sugar with a phosphorus on it is going to be that's called being phosphorylated. And that phosphorylated sugar is what enters into glycolysis. All right. And so that is a review of membrane transport. At this point, you should be able to differentiate between active and passive transport, define osmosis, and predict the outcome of placing a bacterial cell in hypertonic, hypotonic, or isotonic solutions, and identify the types of passive and active transport mechanisms that exist in cells. All right, I hope you enjoyed that, and I'll talk to you soon.